I will be the host for this presentation. And I'm proud and happy to be part of this elite group of readers, thinkers, and answer seekers. This group, the JOC, or the Journey of Civilization Study Group, was started all because of us, like-minded people, getting together 5 years back to read this book, written by Mr. Balakrishnan IAS, The Journey of a Civilization, a travel from Indus to Vaigai. This book actually helped us start understanding a very prominent civilization in a different facet, and also to understand the migratory patterns of people, the language development, the cultural development, and establishment of a civilization. So now it gave us the cravings. So we continued to read another book, The Early Indians by Tony Joseph, which gave us an in-depth discussion into recent genetic analysis of contemporary and ancient populations and evidence-based insights into the human settlements. Now, we as a group got very hungry for knowledge and for answers. So we read another book called Father Tongue and Motherland by Peggy Morgan. This book helped us discuss the birth of languages in South Asia. The discussions helped us understand how mixed languages in South Asia came to life to build an X-ray image of a vanished language of the Indus Valley civilization from the ancient bones visible in modern languages, and also helped us understand the associated migrations of men 4,000 years ago and the Dravidian people and their languages. Now, today, to conclude all these discussions and the material that we have gathered, and some of the questions that have been unanswered, we are concluding it in the way with this presentation by Dr. Raj Mutharasan on genetics and the identity of Dravidian language family. Now, about Dr. Raj Mutharasan, who is the speaker for this presentation.
We can have welcome address now, followed by that.
Yeah. Oh, yes.
Let's have
so yeah, we will have the welcome speech by Professor Arasu Chelaya.
Vanakkam. Welcome to today's talk on the title of Genetics and the Identity of Dravidian Language Family by Professor Raj Mutharasan, organized by Journey of Civilization Study Circle. As the title implies, this talk is special in that it combines genetics and identity of Dravidian languages. Those who already read the abstract of the talk presented in the flyer of this event would have a clear idea what this talk is all about. Hope you all had a wonderful Pongal celebration. Time to watch Jallikattu at least in television while enjoying delicious Pongal. In addition to enjoying Pongal and Jallikattu, now is the time to enjoy this talk. Thiruvallur emphasizes listening to great talks as more important than tasty food. செவிக்கு உணவில்லாத பொழுது சிறிது வயிற்றுக்கும் ஈயப்படும். Here is a great opportunity to listen to the thought-provoking talk. Also, we have a special guest, Thiru R. Balakrishnan, who has kindly agreed to give his remarks on this topic. Once again, I welcome you all for this event. Enjoy the talk by Professor Raj Mutharasan. Thank you.
Thank you very much, Mr. Arasal Chalaya, sir. Now we will, before going into the talk itself, an introduction on the speaker, Dr. Raj Mutharasan. He is a retired professor of chemical and biological engineering from Drexel University in Philadelphia, US. He did his undergraduation at IIT Madras and graduate studies at Drexel University. He has published extensively in his research area in biochemical engineering and biotechnology. His passion includes ancient history, genetics, literature, and photography. Today's talk covers one of his interests on population genetics. Welcome, sir. We are all waiting to listen to your very elucidate and thought-provoking talk.
Mute
Unmuted yourself.
I'm now unmuted, right?
Yeah.
Okay. First of all, thank you very much. And vanakkam to all who are here. And of course, the description given by Shubhadra suggests that I got to perform today. Glad to share with you what I have learned. And I want to tell you at the outset, I had given a talk such as this a little bit earlier, about a year ago, and in this group. And of course, that has gone through a lot of discussion, as was described by Shubhadra earlier. And about 2 months ago, due to Balakrishnan Iyer, he now heads up this research center in Chennai. He invited me to talk about this topic because he had listened to my earlier genetics talk. And so I've translated that title into Genetics and Identity of Dravidian Language Family because that research group, that organization is more language oriented. And as all of us know, people who speak a language, as they travel, they take the language with them. And as they interact with another language group, as the consequence of interaction, languages evolve. And of course, there is a genetic component in language, same as how the opposite would be true. So I gave this talk about a little over a month, 2 months ago via Zoom. And so this is a rework of that same talk, but I've added a lot of features to this because in that talk I was constrained to 20 minutes. Well, now I have a little bit twice as long. So this talk is approximately 45 minutes. It'll be heavy on genetics and light on the linguistics because that's a very large topic. But I'll give you a flavor of it so that you can read the text that all of us read over a 2-month period, which really provides enriching experience into how languages evolve. Anyway, with that sort of an introduction, let me go to what I have outlined for my talk. First, I'll give you an elementary introduction to biological foundation of ancestry, which consists of 2 parameters, what is known as, I'll define them, I'll describe them as we go along, what is known as Y DNA haplogroup and mitochondrial DNA haplogroup. And then I will give a brief outline on how ancient DNA revolution has really kicked up a huge sort of information on how our ancients lived, where they came from, and how they traveled. And that story is less than 25 years old. It is not a long story, but I'll point out key important elements and then get into, because of these new tools that have come into being, we've been able to articulate very carefully with scientific evidence the 4 ancestral strata of Indian genomes, primarily 4 ancient strata, what they are and how they came about, when they came about, how this was determined genetically is what I will share with you. I won't get into the nuts and bolts of the details of methodology, but I'll be able to describe it in a fashion that you'll be able to relate to how these kinds of information came to the surface, if you will, through. I'll rely primarily on published peer-reviewed journals. I don't like to get into speculations as far as I would avoid. When I try to provide an opinion, I'll earmark it. But remember, data never lie. It is only the interpretation that could be different, right? And then the chronology of genetic admixture of these four ancient strata that India has experienced, I will get into that a little bit of a detail. And then I want to bring all of these theoretical ideas down to earth. There are three individuals who are currently listening to this talk who actually went through a genetic analysis using commercial entities, and they allow me to share their results, and I will share their profile, and I'll give you validity to many of the theoretical discussions that are presented in the journal. So that will bring down to earth all of these ideas. And of course, I've shared my own genetic profile earlier on, and I won't get into that here. And then finally, get into the linguistic aspect. Since we did about 2 months of study of Peggy Morgan's book, it just brought in a new, her view of linguistics is different from the classical linguists who work with words. She works with syntax and grammar that provides an interesting insight into how Indian language is developed. And I'll give you a flavor for it. It's a huge topic, and so that it could think about that as a stimulating factor, if you will. Okay, elementary biology of genetics, if you will. We all have trillions of cells in our human body. Every cell harbors one copy of chromosome. Chromosome is like a memory cell in your computer, which is then converted into molecules that then may enable you to function. Of these, there are two things that we need to keep focus on, which we call as ancestral DNA. This is Y chromosome, which is present in all men. Women don't have this. We have 23 pairs of chromosomes, out of which 22 pairs harbor the genes corresponding to creating the molecules that are required for us to function as a human being, as an entity. One is called the sex chromosome. Females, women have X and X, while male have X and Y. And the Y is passed on from father to son, son to all male lineages, while the mitochondrial DNA, which is present in multiple copies, as much as 1,000 or 10,000, particularly on cells that do muscles, if you will, which are high energy cells, there are thousands of those copies, and they are passed on from mother to all her children, both male and female. So you can think about the Y chromosome as a male and paternal ancestry, and the mitochondrial DNA as a maternal ancestry. Besides this chromosome, the 23rd chromosome, there are 22 other chromosomes which biologists call as autosomes, the pairs 1 to 22. They contain genes. These are entities that enable us and produce protein and hormones and various different biological molecules that enable us to function, if you will. And that gives you their total ancestry. So a person may be of a particular ancestry via Y and via mitochondrial, but may harbor genes from an entirely different ancestry. So looking at autosomes is a very important aspect of understanding our genetics, if you will. Unfortunately, determining a Y chromosome is easy. It's a very easy experiment. Determining mitochondrial DNA is also very easy experiments. Getting the autosomes is a more complicated essay. It's a more laborious essay, and it's a more expensive. So most people, until say 5-10 years ago, did only the Y and mitochondrial. Only in the last 10 years, we see a more broader picture of autosomes come to the picture. And I'll point out to the stories. And I also want to enable this, make this talk to stimulate some of the young people in the community who are data scientists, because many of the software that is used in this field are public domain and they're available. I'll point out links to them. and many of the ancient genomes are public domain. You can download them and actually do calculations on your own. So hope some of this talk will stimulate a few of you to look into more details. And the last slide will point out things where we like to do work on, because being a researcher myself, whenever you study a particular subject matter, you figure out there are questions that are unanswered. Okay, this is the biology part. Now, the geography is important for us to know. So this is a sketch where I will be referring to some of these areas. And this particular area, where is my...
Yeah, your pointer is missing here.
Oh, there it is. Okay. Okay, here is my pointer. Hope it is sufficiently visible. This is Indus Valley. And of course, this is India. We won't get into this aspect. These are much later time frame. We'll be referring to BMAC, which is Bactria marginia archaeological complex, which is a large complex in the outer edges of Afghanistan. In popular literature, it'll be called Oxus civilization, which butts against, and many people who came from this area, the steppe people, if you will, this is Caspian Sea, they came via this way. They didn't stay there, they came through this way. And Saracens are people, it's another ancient civilization. We have known as period 1, period 2. And those folks had pre-metal, post-metal civilization. Pre-metal folks had connections to people in Indus Valley because we have found many of the jewelry that was produced in Indus Valley out there. So they seem to have traded, and they're genetically connected to Indus Valley. That I'll get into the details of that. Similarly, people who lived here were genetically connected to Zagros Mountains. You'll see the three examples I'll show you. Their great-great-grandfathers lived in this region, and of course lived in this region too. But anyway, these are for you to get a feel for this area. And Swat Valley is another area in northern Pakistan. We have ancient genetic relation data from this particular community as well, although later though. Now, getting back to Y and mitochondrial DNA, this gives you the picture of the ancestral. So the Y is classified as major branches of A, B, C, D, and so on and so forth up to Q. These are major branches. Within major branches, there are sub-branches and sub-sub-branches. You can think about a tree trunk being, say, L, and then a branch would be L1, another branch would be L2, and then within L1 and L2, there may be A, B, and so on and so forth. So these are major branches. And for example, LM37, which is an important marker for Indus Valley, is major branches M. Within that, the mutation that occurred labeled as M27 would be called L1, for example. And then of course, these folks migrated into Indus Valley and then further migrated into mainland India. And similarly, mitochondrial haplogroups are labeled L, M, and so on and so forth. And they will have a sub number, M31, for example, is a mitochondrial. The M is the major haplogroup in mitochondrial DNA. M31 is a sub within that sub branch of that M haplogroup. M31 is something that you'll commonly find in India. That is why I label that. And then the third piece of heritage, not easily often found, is the remaining part of the chromosomes 1 to 22, that harbor about 20,000 genes. There are a lot, that's only 1% of that particular molecule, but the remaining, we are still learning what they harbor. But there's a whole series of segments in there which we do not know that have a contribution. They are simply blanks, if you will, at this point in time. So as time progresses, we'll discover what they are, but not all of that isn't. So there's a large part of that domain is still unknown to some extent. Large part of it has seemingly irrelevant material too, something that we need to keep in mind. Anyway, this is a growing field. Keep that in mind. So a quick outline of historical timeline on this. The idea of out of Africa model was proposed in 1984. Stringer is generally attributed to that. So it's within our lifetime, for most of our lifetime. It is well past my college days. 87 was the discovery of the first mitochondrion, if you will, maternal line. That is attributed to Wilson's lab. 1991 was an important part where Professor Cavalli, I wouldn't be able to pronounce, I'll call it a CS, Professor CS from Stanford University launches this Human Genome Diversity Project. And he sampled all over the world and assembled sort of the Y haplogroup network, if you will. So that body of knowledge really evolved in the 90s. And then, of course, the discovery of one of the most ancient Y haplogroup M130, which we find in India, by the way, was discovered by one of his postdocs, Underhill. And of course, in 2008, we discovered that in Madurai, near Madurai, Theni district, Andi Devar, I think is his name, Virumandi Devar is his name. And that was that discovery is attributed to Professor Pichappan of Madurai Kamaraj University, and that is in part due to Spencer Wells, who was in the same lab as Underhill was at Stanford University, but he went to fieldwork. Anyway, the Y paternal approach really evolved after 2000, and of course in 2009. At the extreme other end of United States, David Reich at Harvard, he did his PhD in zoology at Oxford, but then got engaged in Whitehead Institute, which is on the campus of MIT. That's exactly where Professor Corona, a Nobel Prize winner of Indian descent, also had his lab. And of course, he passed on now. I think he passed on about maybe 10 years ago. But he and Corona might have interacted because this would have been early 2000. And then he went on to take up a faculty position at Harvard University, and he launched on to the Denisovan. On the other hand, on the other side of the ocean was Svante Pablo, who won the Nobel Prize for putting together Neanderthal genes. These names would make a greater sense, as I point out, because they lived in India. Both of these species lived in India, as we know from a genetic, and that body of knowledge really evolved in the last 15 years. We did not know this prior to that. Okay. Got to be, okay. Now, ancient genome really provides a rich set of information on our history. And the field is very young. It's only about 8 years old. We had just one. The first one was around 2015, and then it took off. And of course, this is the latest chart I could find published because Nature published this because it celebrated the hallmark of reaching the mark of 10,000 ancient genome. Bulk of them were from Europe and Russia because of cold climate. They were well preserved. And what we find in the rest of the world were far less, only the 1/3. And of the 500 in Central Asia and South Asia, only one was found. Of course, Rakhi Garge lady was found in India. And hopefully in future we'll find more. The more updated, by the way, for those who are data scientists, the link, I will make this slide available to the community so they can look at all my references. Many of them are in open access. The link that I've provided here will give you access to the ancient DNA database. You can download them. They're all SNP files. I'll tell you what that is. And then you can do your own work with them using software that's also downloadable from Reich's lab, and which is probably my next slide. Here is, I was trying to get an update on what is the status of ancient DNA, which is now in excess of 12,800, out of which 2/3 of them are from Europe and Russia, cold climate. South and Central Asia is about between 600 and 700. Most of them are Central Asia and Iran and Afghanistan. Very little from India, just one or two. And East Asia, more of China and Southeast Asia, and Americas and Oceania is about 1,000. And I've listed here just for a picture, screen capture, tell you what the data looks like, which is really SNP data file, which tells you on what chromosome, what SNP is located, a particular molecular structure that is important in analysis of DNA, if you will. Now, India has been populated by hominins. These are not Homo sapiens, but our ancestors, Neanderthals and Denisovans. And they've come around to India for a long, long time, a good million years. This is documented via the residues of their occupation that we find, namely stone tools and specialized. These are fields that people have. One of them is very near Chennai at Adirambakam, about 40 kilometers from Chennai. Some of you may have visited them. That's 1.5 million years old, according to what it has been dated as. But the one that is well recognized by UNESCO is the one in Madhya Pradesh, Bhimbetaka. I've visited that site. That is also goes back to Lower Paleolithic, that means very long ago, several 100,000 years ago, occupied by pre-Homo sapiens. They were Homo erectus, and they have left residues of stone tools from which people have thought about when they lived. And of course, you find them in Rajasthan, you find them in Madhya Pradesh, you find them in Andhra and UP as well. The UP one has got a much later, more Neolithic, if you will. And India has been home for why that is the case, because as you go farther away from equator, it is colder, and not having clothes, perhaps ancient folks moved towards where it was warmer. That might be the reason. That is my hypothesis, but not necessarily a proven fact. Now, going to Y haplogroup, it's a very large map. I've given links at the bottom where you can get the entire map, which is a giant map. What I've done here are the important Y haplogroups, that is for the Indian context. The red dots are the ones that you want to, that we find in abundance in India, C5, if you will. M130 is one of the most ancient ones found in India, which is Virumandi Devar in Theni district. His siblings, if you will, his descendants, C1 through C5. C5 is found in South Asia, namely in India. You find them more in tribal population than in the general population, but they are found in all communities, if you will. D is a descendant of the original out of Africa group, which is labeled as CTM168. That is the designation of that particular molecular structure, if you will. And one of those is found in Andaman Islands. These folks also moved north overland, and the Ainu people are relatives of the Andaman people. They're of D1A2, if you will. And the folks that you find in Andamans are D1A3. Remember, D is the major branch. 1 is the sub-branch within that. Within that is the sub-branch A, and within that the category branch 2 and branch 3. So you can think about that as a large branch branching off into smaller branches, which are the labels 1A3 or 1A2. And then corresponding to from the original inhabitants where F was formed, and you find them in India, particularly in tribal groups. And from that, H was formed, and the labels that follow these major branches are mutation numbers. And you'll find, depending upon which literature you read, some of them will be mislabeled, so you have to be careful. Some of them will be correctly labeled, so you have to keep that error being present in the body of literature in this field in the back of mind. And these gave rise to I and J. I went into Europe. J came into South Asia, if you will. You find J in Iran. They also populated Indus Valley. And of course, K came from the F, and you find them in India. And of course, L came out of that as well. And of course, as you go down further, R is a major group that was founded in Central Asia. They migrated into India, R1A, the dominant, you find them a lot in North India. You also find them all the way down in the south. This is what people have called a steppe migration. Out of R came R2, but R2 seem to have distributed themselves from Central Asia much earlier. You find them in BMAC, if you will. You find them in Iran. You find them in Indus Valley Klein. And so R2, although R1 and R2 have the same ancestry, they seem to have left Central Asia via different paths. We don't have solid evidences of the trajectory, but we have really solid evidences of how R1A traveled because of ancient DNA that's available. This is a complex map, but let me go on. Since I'll make this available to all of you, you'll be able to dig into this much more detail, if you will. Now, I've simplified this into three major groups. I've shaded them as light gray, slightly darker gray, and darkest gray, if you will. The first three are the most ancestral layer that you find in India: C, D, and F, and K, and H. You also find them in, by the way, in eastern edges of Iran. And then the layer which is L, R2, and J2. This folks, in fact, the light gray folks and the dark, slightly middle gray folks joined together and founded the Indus Valley. That's a hypothesis, but that hypothesis has been proposed by many people in the field, if you will, scholars. And later, we have the R1A migration, which is the steppe folks migration. We know the trajectory, we know also the time, and we also know that they came in dribbles over a 1,000-year period. It was not a single movement. And that is based on genetic evidence. I'll share that results with you. And then the Austroasiatic people who came from current day Myanmar, Thailand, Vietnam, came into the East Coast. They also went up in the Northeast, and you find them integrated linguistically, both linguistically and genetically, with people in Orissa, Bengal, Assam, and that part of the region. They don't seem to have come down further south. At least that's what genetics shows us. So my choice of this gray color is the lighter gray, this is my opinion, are the darkest complexioned people. The light and the medium gray people are people who are not as dark but were lighter. And then the darkest colored people were the most light-skinned people, if you will, in terms of. So if a person has, it doesn't mean if you're light-skinned, you belong to one group or the other, because that particular gene may not be expressed. And that has been very well studied. I'm saying that in a joking way, so don't take that too seriously. But there was a complexion difference, if you will, in the group. Okay. Now, this is the mitochondrial DNA.
One session. Yeah, someone is making annotation. Can you please not do it? I don't know who is there, but someone is doing it. Can you please
Who
not?
is doing it?
I couldn't find who is doing it, but
Okay.
thank you.
Okay.
Yeah.
Okay.
Bama, anything unusual?
It's annotation. They are marking it as Aya is talking. They are marking it so that is visible to us as well. Like you can see one yellow mark and one blue mark. That's
Oh.
not. Yeah.
Oh, that's not Dr. Mutharasan's.
No,
No.
no, no. He is not doing it.
No, mine. This is mine.
This is cursor is that tail.
Oh,
Yeah.
okay, I'm not surprised. Okay, carry on.
Okay. So far I've told you the overall layout of the Y chromosome. Now let me go to the mitochondrial DNA layout. This is a complex map, but it's available for you to dig into when you have some time. And my purpose is not to spend time on this, but rather simplified it for the Indian context. So the people who came out of Africa, the early migrants, were from East Africa, were L3, and what we find, they diverged into M and N. These are two major branches, and you find them in India. And M branched out into M1, M2, M3, etc. The boxes that I've put here are the mitochondrial DNA group that is found in India. There are other M's that are found in East Asia. And most of this mitochondrial DNA that is listed here are found only in India. You don't find them elsewhere. That's an important point to remember. And similarly, the branch N divided up into R and U, and R that I've listed here are mostly found in India. There are other Rs that are found in East Asia, and these Rs primarily are found in India. Similarly, U1, U2, and U7. U1 is not found as much in India. U2 is found. In fact, one of the scholars in this field calls it as U2I because all its sub-branches, I, in his opinion, was India. All subscripts of U2 are found only in India, but U2 is also found in Indus Valley clan. I'll point this out. U7 is found in India, not as much. U1 was recently discovered in India in the Koraga tribe in Karnataka, but it's very rare. It's primarily found in Iran. So this is a simplified version, context for the folks who live in India, if you will. Oops. Now, ancient migration routes as established by Spencer Wells' group and the Stanford Research Group points out that the ancient folks from Africa moved into Arabia along the coastal route and then all the way down to India, where you find C M130. And then they migrated along the lines, along the coastal route into Australia. And the derivatives of C are found in Australia. And now, remember, in ancient times, I've listed here the Ice Age, 115,000 years before, down to 12,000 BCE. So the distance with the coastal route, the sea level was almost about several hundred feet lower, and the distance between the mainland India and the Andaman Islands was only about 30 or 40 kilometers, something in that order. So they might have just sat on a log and moved over. And they remain isolated as the sea level rose, and therefore very isolated population of the world lives in this Andaman Island, particularly Sentinels on the northern edge of the Andaman Islands. So Andaman Island harbors some of the most ancient first Indians people within this community. And the discovery of CM130 in Theni district in 2008 by Professor Pichappan's group, and of course that was under the auspices of National Geographic Society project headed by Spencer Wells, was a confirming step of the trajectory of the coastal route. They had previously discovered sea in Ainu in the Japan, and therefore they knew the overland route existed. They knew the sea was here, but they couldn't fix how they came from here to here. And the fact that they observed this was an important discovery, if you will. This one discovery is attributed to Dr. Tangaraj of CCMB of Hyderabad, and they attributed this to Professor Pichappan. So the Indian scientists have had a major contribution to figuring out this ancient trajectory path, if you will. Now, I'm now coming down to what genetically is supported. The previous one is spot points, now more solid evidence. So the early trajectory is what I showed. Now we go back 10,000 years, only 10,000 years. We know, I'll show results of this, Iranian farmers migrating into Indus Valley and then further into Deccan, about 8,000 years. BP stands for before present. And that is established using genetic evidence. And we also have genetic evidence of deposits of carbon, ash mounds, as you will, in the Deccan. There are two sites in Karnataka, one in Andhra, that is dated 4,000 years before BC. So they were contemporaries of Indus Valley civilization. And then we also have genetic evidence of steppe folks who came into India via Hindu Kush, And that's an important distinction that came out of ancient DNA analysis, because before that, prior to 2009, most scholars believe the steppe folks went via the Iranian plateau, the left hand, if you follow my trajectory, that they from Caspian Sea, they came via this into Iranian plateau into Indus Valley. That has been rejected now. And that it is very clear that the steppe folks went east and then dribbled in over 1,000-year period via Hindu Kush through the mountain passes, Karakoram Range, into Indus Valley, into the northern region, and then into Gangetic. And they seem to not go farther than the main UP region, if you will. And then they dribble later on into the far eastern part. And now, Austroasiatic people, again, by using genetic evidence, we know they came via overland route into this region, but they also seem to have come via sea route into the eastern region of India, Odisha, if you will. Now, this is again based on genetic evidence, and I'll share results of how they came into this result. Now, what throws into the sort of a monkey wrench is Chenanur excavation result that was published last year by Tamil Nadu Archaeology Department, which is near Tiruvannamalai, where they show using solid archaeological evidences that the earliest settlement goes back to 8000 BC, that is 10,000 years ago. That is a contemporary of Mehrgarh, which is the first settlement in the Indus Valley region, and a continuous evolution of culture there, because they see the ancient bones, ancient bangles, if you will, made out of bones, etc. And then Stone Age culture evolving into a metal culture. And so the 4 layers that they've been able to show, they were continuous and then abandoned about 1000 CE, is a very rich new information that has come to the floor. And I'm sure there'll be more work that'll be done over there. It didn't evolve into an urban civilization. Remember, I want you to keep at the back of your mind, early on we were hunter-gatherers. We just lived off the land, if you will, what was there. Then we invented farming, and farming means you got to settle down. So settlements began to grow. And of course, you would also hunt at the same time for getting nourishment. As settlements grew, there were excess farming. You have more food than you consume. You trade. As a consequence of trade, you move. And of course, you have free time to make, bangles and jewelry and etc. and sculptures. And now that, as that specialization occurs, you now need organization in the society, and therefore you evolve into urban society, if you will. And so urban society is what we call a civilization. Settlements are not considered civilization. And so evolution from settlements to civilization occurs as the community becomes more creative in the way they apply their resources and create excess free time to create all kinds of artifacts that they can then trade and become right. I want you to keep that at the back of mind. So there's a big distinction between a settlement and a civilization. I'm a little bit long-winded. I'm sure I'm going to run over time. Now, this illustration is a little simulation I ran to give an illustration of how scientists arrive at try to figure out when one group of people blended with another group of people. So this is a simulation. Think about blue as one group of people of a particular genetic characteristic, red as another group of people having another genetic characteristics. When they mate, they produce children, they would have the blend of these red and blue chromosomes, if you will, which I've illustrated as second generation children having this. As they mate, as these two groups mix more and more, on the left-hand side, I have generations. 5th generation, you find same amount of red and green, but they're distributed in different sizes. As you go further down to 100 generations, the segments that are blue, segments that are red are smaller, but they persist. If you add up the total red and the total blue, they'll remain sum up to the same 100%. On the right-hand side, I have years before present. If you take an average generation time as 30 years, and that is what this calculation is, 2 generation is 60, and 100 generation is 3,000 years. And that puts us within the time span of people migration, right? So this is the tool researchers have used to establish when the Austroasiatic speakers came into Odisha or when they came into Nagaland and the northeastern section, if you will. In fact, more important for us is the migration into Odisha, where Bala Iyer has served many years, so he knows a lot more about that culture than I do. One interesting point I just wanted to share, they don't have gender in their grammar. And of course, we have gender in ours, she, he, and it. They don't have, and the language they speak, Odia, they don't have gender either. And of course, this comes up as part of the linguistic analysis. Let me not, there are a lot of interesting stories to share, but I don't want to be too much distracted now. This is the model, this particular group, Murjani, she worked in Professor Wright's lab. Now she set up her own lab at Berkeley. This publication goes back almost 15 years. They looked at over 70 communiques. So we have arrows here from somebody else and stars, decorations, but those are not mine. Anyway, we have, I've divided that table contains about over 70 communities. I've just simplified this table for our conversation. So age of step generation flow into India. And so using the recalculating, it's a mathematical method of what I had shown in the previous slide. They were able to recalculate when these two groups, namely the ancient Indians and the steppe people, blended, if you will. So the incoming people, according to many scholars, were mostly men, and so they just took on women who were present in India, and they created daughters, if you will, and children. And they go back on the right-hand side. Their calculation, I think they use generation time of either 29 or 30. You find that to be in the orders of 2,000 and 3,000. And so this gives us the folks who came from steppe, they dribbled in. They didn't come in a whole slot, but they came over 1,000-year period is the current estimate. They might have continued to migrate as it happened during Mughal time, if you will, and other times. But it took place over a generation, multiple generations, and they blended with the people who were already present. And of course, you find they're blending with different community groups for different, if you will. Remember, there are underlying assumptions in this calculation, the generation time, and of course, the sample, the red and the green chromosome I showed on my previous slide. For example, for the Madiga group, they would have taken 3 or 4 people. Okay. 3 or 4 people and use this as a model for them to calculate as the result of how they came about. They would have taken the Onge group Andaman, they'll take the steppe people genome, and then from that they'll try to calculate when the current population of genome of that particular community came about. So it's a mathematical calculation. I'm sure it has errors and it's estimates, but it gives us a ballpark figure. That is the important part that I wanted to carry forward with. Now, this is not my favorite slide. You'll find in that particular slide, we have ancient North Indian, ancient South Indian. I would urge all of you to read chapter 6 in Professor Reich's book of where we came from, or the chapter, I think it is the second or third chapter of Tony Joseph's book, where the description of these two terms come about and how they pretty much point out they were political signatures rather than scientific signatures, because there's nobody called ANI in India. There's nobody called ASI in India. They're a blend, mathematically created genome to communicate that every Indian is ancient Indian, if you will. And so scientifically, these are not proper terms. And I've included here that ANI is formally, mathematically, 70% IVC people and 30% steppe people. Remember, steppe people were not native to India. They came into India about 4,000 years ago. And the ASI has been designated as mathematically 25% IVC people and 75% ancient South Indian, if you will. This is the Onge tribe in Andaman Islands. So these are theoretically created genomes to communicate a political message rather than a scientific message. That's my opinion. I want to make that point. But anyway, this continues to exist, unfortunately. But some scholars have gotten away from it, but I'll also point it out. Okay, now we come to the serious migration of folks from Indus Valley. I'm approximately 30 minutes into my talk. I'm not sure I'm halfway through, but I hope you'll give me the extra 10 minutes. Now, the haplogroup L, until this paper came about, they looked at ancient genome results as they came into being and then used a mathematical model known as a Bayesian dating analysis. This is a network analysis Bama might be aware of. This is what one uses to analyze networks, if you will. It is a statistical, a mathematical model of figuring out how different things get connected, if you will. So here the data are the genomic data, and then how they moved about, if you will, is calculated theoretically. And this particular paper that came out about 18 months ago points out that the origin of L haplogroup was about 20,000 years ago in this region of Mesopotamia. Remember, it uses Zagros Mountains. It is at the far end of Indus Valley, if you will. This is the region. And then about 10,000 years ago, they expanded into different directions. They did move towards India, Indus Valley. There's evidence of it. And then migration to South India is about 8,000 years ago. We have genetic evidence for it. They marched along. And of course, 4,000 years ago, they populated a very large fraction of it. This is only L1M22. And the sub-brothers of them are M27, I think, if I remember right. But L1 is an important major group that came into being. Now, how did they discover this was based upon, this is the network. On the left-hand side, the dots are the ones that are calculated using Bayesian model, using ancient DNA as markers to check their accuracy, if you will. So it is based on data, but the dots are based on a mathematical model. We don't want to look at this entire network, but the part that I want you to look at are the part which consists of blocked about, I think it is 165. This is based on 165 Y haplogroup for people in various different regions. The region that is of interest to us are the people who are marked in red because they're in South Asia. For example, that's in the southern end, the bottom end of this slide, which is the L1 haplogroup, which is out of here, L1M22. They branched on to the L1A, L1A1, and so on with a mutation M27 and M76, and they branched off into India. On the right-hand side, who are the current day people, 165 people. You find them in Balochistan, Brahui-speaking people, Sri Lankan Tamils, Punjabis, naturally, that's in Indus Valley, Telugu speakers, Indians in Singapore, Sri Lankan Tamils, and Telugu speakers, and so on and so forth. And you find a fair number of Gujaratis as well. So it shows this L1 has a connectivity to South Asia. That's an important message from this particular slide, if you will. I want to take another example of this. I took this example because Gujarat is very close to Indus Valley. In fact, one of the important Indus Valley sites is in Gujarat. And this is only about the tribal communities, 7 or 8 tribal communities in Gujarat, which is listed here. I won't try to name them. And on the horizontal axis are what their haplogroups are. They determine only the haplogroups. And of course, these authors list exactly what mutations they actually measured. That way you can double check against the global Y haplogroup designation. The important thing to remember is you find a number of them are C, F, H, H1, H2, which are ancient layers of India. These are among the first Indians to come into India. And of course, you then see Indus Valley folks like J2 and L1, and of course, R2. They're also there. And of course, you have R1A, which is the steppe folks. R1A is about 18%, which I have labeled here as 18% of their heritage is steppe ancestry. 82% are pre-1900 BCE, which contains both parts of ancient Indians and the Iranian farmers, if you will, who are people who formed the Indus Valley civilization. Now let me get on to mitochondrial haplogroup distribution. Now this study was done, there are two population here that I'm listing here, which is here, genetic admixture of evolved in 21 communities of focus only on Tamil Nadu in South India. It's a publication going back almost 15 years. So they didn't have high resolution of the mitochondrial. They did the broad categories of M, N, and U. They didn't try to go down below the subscripts M35 or M31, etc. Probably those microplate arrays were not available at the time and they were yet to be discovered. And so this is an ancient one. The second one that I wanted to list is because they include primarily another group of people, which included the Brahmin community, Iyengar and Ayers, and that was even earlier, 25 years ago. They also didn't have the high resolution microarray plate, so they just looked at M and U only. They didn't look at N, which is upstream of U. And so when you look, the main message I wanted to, you can dig into this later on since I'll make these slides available to you. The big message you get is the macro group M dominates this. M is one of the most ancient. So pretty much all of these folks, various different castes, various different communities, Anglo-Indians, North Indian Jains, South Indian Jains, if you will, Tamil-speaking Jains, predominantly they are children of ancient mothers who lived in India 60, 70,000 years ago, predominantly. And of course, N and U are also present in this community. And now if you look at, and I was in search of M2, which is the most ancient mitochondrial haplogroup. And then of course, in my own private conversation, I call them as the Mother India because they were the first mothers. We have not discovered M1 in India yet, but M1 theoretically exists when we looked at the structure of mitochondrial DNA. M2 exists. And 100% of Kuruman, at least, by the way, this data comes from Archaeological Survey of India published 2021, recent. And they looked at some 70 or 80 communities in India and they published them. They're available. It's behind a paywall, unfortunately, but I was able to get this information through my library. So M2 varies considerably. In the tribal group, usually Kuruman, 100% of them, of the sample they tested, if you will. I must make a remark here. None of the sampling that has been done by any of the studies in this population by Indian scientists or elsewhere were done statistically representative. That's a particular terminology which would be meaningful if you know statistics. I don't want to get there. They were always taking people who were willing to give sample and who were unrelated. Those were the two criteria they used, and therefore we don't have statistically representative sample of any of this population. That's within quotes, and that is the nature of data in this field, but we have to live with this. So Todas are 75%. These are Nilgiri people who live in the Nilgiris. And so isolated communities harbor some of the ancient features of first Indians. That's my hypothesis. So studying them is an important job for all of us. And I hope that would be taken up. Not enough has been done, and I'll point this out in my last slide. If you now take another project that was done about before ancient data came to the fore, they looked at 1,000 genome across the entire world, looking at a mitochondrial DNA. And of course, they looked at East Asia, Europe, India, and of course, various different parts. The part that I want us to focus on is they looked at Bengalis in Bangladesh and Gujaratis in Houston, Indians and Telugu speakers in UK, Punjabis in Lahore, and Sri Lankan Tamils in UK. And so all of that data is contained within here. The Indians are mostly M, there are R, and of course U. And the rest of the haplogroups, which is on the vertical axis, are very little. Most of them belong to, in fact, all those who are listening to this talk, you're very likely to be falling into this category, if you will. M, R, or U. The probability is your mitochondrial DNA is M is about better than 60%. Now that's an estimate based on my hunch numbers, looking at various different data. Now, I want to quickly look into Raki Gargi. This is the one genome that we have available for us to give us a clue as to what the people who lived in Indus Valley actually looked like. But of course, we have a lot of data on people who lived near Indus Valley from the Zagros region and of course further north in the Tajikistan region, but not hardcore Indus Valley. She was a female, about 5 feet 4 inches, something in the orders of about 35 in the age of 35. She was buried with a lot of some grave goods, pottery, if you will. There's a little bit of jewelry. She was not of a high esteem. She didn't have high quality jewelry, but she did have jewelry on her. She was buried in the north-south domain. And her mitochondrial DNA was determined as U2B2. U2B2 is something that you find, and I have a little box here on the left-hand side. You find 23% of the sample that was taken from Urular community. Urular are the dark-skinned people in the Nilgiri district. They're also found in other parts of the mountain. 23% of them contain the same they belong to the U haplogroup. This was not a high resolution, but you find U2 quite commonly in South India, if you will, along with the M, if you will. Now, her genome was analyzed, I should remark. The genome is a very large molecule. There's a side step. And of course, typically there are millions of SNPs. That's how we define a genome mathematically. But scientists have discovered out of that, about 1 million would be a very good representation of an average person. And when not all of her genome was available, we got only about something in the orders of about 50,000 SNPs out of her, which is statistically representative of her whole genome. That is how it is to be taken. As I would point out, they've been trying to get the ancient genome out of the skeletons from Kiridi. My understanding from press reporters, they've been able to get only about 5,000 SNPs, not 60,000. So they are in that process. Hopefully they'll improve. And if they are able to get 60,000, we'll have ancient genome from South India. That is yet to be achieved. That would be a major mark in this field if and when that is achieved. I can get in discussion of why that requires attention, but I won't distract ourselves from here. But the key thing to point out is she was a mixture. She had a substantial amount of Andamanese hunter-gatherers, but she also had a substantial amount of Iranian hunter-gatherers, if you will. I have specific numbers on another table later on. Now, I include this slide to give you an indication of what Indus Valley surroundings look like. Okay. The reason is we use that as a model to figure out what was an Indus Valley civilization. On the left-hand side are the Indus Valley ID or rather ID of these ancient genome, because if you go into Professor Reich's database, you can use that label and download that entire SNP file, and then you can go ahead and that universal ID is very helpful if you're getting to do any analysis. So Rocky Gargi lady in the data science world has got a name, which is I6113, if you will. Now below that, our Shokta, or I wouldn't be able to pronounce the name, is in the Zagros Mountain region, and those labels are available. And you'll notice that the mitochondrial DNA was M30, which is closely related to the M's that we find in India. But the mitochondrial, the other person was R7, but his Y DNA is R2. The R2 is found all through India, if you will. And now Gonur, which is in BMAC, is H1A. This is like ancient, really ancient, like 60,000-year-old. This is the AASI type of heritage. And he was in Gonur in BMAC region, if you will. And of course, mitochondrial was R2. R is found in India. I'm not sure whether R2 is found in India, but R5 is found in India, which I know personally. And another location here, male, J2A. J2A is found commonly in India at about 5-10% level, all the way down to Tamil Nadu. And of course, Sarzam, another neighbor of Indus Valley, Y was T1A, not common in India, but U7 found in trace quantities in India. And then Swat Valley, which is a later, which is 2,800 years before, R1A, which is a step ancestry, with a U2B2, which is identical to our Raki Gargi lady, the only Indian lady whose genome we know as a part. Now, going back to the right-hand side, we know the age of these, what these particular skeletons came from. And then her genome, entire genome, this is estimated, if you will, was 1/3 consisted of AASI, the references, the Ongi tribe from Andaman Islands, and 64% Iranian Neolithic genome, if you will. And the rest are given here. Now going to Swat Valley, which is in northern Pakistan, is 24% AASI and 56% Iranian Neolithic. And the remaining would be, my guess would be step ancestry, step autosomes, if you will. So it gives you a flavor for what Indus Klein looked like, right? Now, both of these papers at the bottom are available in open access. The important point, the data scientists in this field, you want to download table S1 from this site, which contains the data that I'm showing here, the entire SNP file. If you want to fool around, look at different ancestry, you'll be able to do that using software that's also available at David Reich's website. Now, what does ancient DNA show? This is sort of a quick summary out of Narasimhan's paper, which preceded Vasant Shinde's paper by about a year or thereabouts. Primarily you find there J2A, L1. I already told you the story of L1. And then R2, another ancient Y haplogroup. And you find them all the way into the mainland India. So the periphery contains the genome of people who are now currently living in India, if you will. Now, why this in current distribution? This particular study was interesting because it focused on Chennai. There are many of you in this listening who are from Chennai. And so these authors looked at people in Chennai. The nice thing about this study is they just took 145 people. They didn't divvy up people based on caste or religion or etc. They just took up 145 unrelated people who are living in Chennai and then looked at what their Y haplogroup is. It was a low resolution Y haplogroup. They also looked at people in Karnataka and also in southern Maharashtra. I'm not going to get into that. I'll just look at the Chennai data. What does that tell us? Now, I've taken the data and assembled this left-hand side table. 5% of the people in Chennai are the ancient folks. F, G, and H, and J2 are and L and Q. And of course, Q is an outlier in my opinion. And of course, R1A, which is 18%, and R2. If I were to put that into this green box, we have three layers. You know, the early slide I had, four major layers of ancient genome. Three of them are in Chennai. We have the deep South Asian lineage, which is H, F, and C, and which is about 35%, and about 45% are Iranian IVC layer, which is L, J2, and R2. And then 18% are step-derived genome of RNA, if you will. So you have, out of the four, three are represented in nice proportion, probably in proportions of this. I have a refinement to this with a more recent data, which would be interesting too. Okay, I must include this because this was one of the early pivotal papers that shed a lot of light on the Indian tribal population, much due to Professor Pichappan's work and Spencer Wells' work, National Geographic Society project. They looked at only the tribal and the communities, and the data is presented on the basis. It's a very large table. It's downloadable. It has errors, so you got to be watchful. I've written about the errors in them, and I think they might have been republished that. But the key thing that I wanted to point out of this was I was trying to seek some information as to the nature of the tribal population. So taking his data and parsing it out and taking L as a proxy for Indus Valley, what I find is most of the people in this tribal population do not have the L ancestry, if you will. So that is one conclusion I came to, and this is based on only this particular set of data. I'm sure there are data that contrast this. So the story that I want us not to forget is the tribal population may be harboring certain features of maybe cultural features of ancient Indians and maybe ancient languages. I'm not sure, but that's a hypothesis to be tested. Now, these are further details of the same set of slides. I looked at Irulas. They are primarily the ancient Indians, if you will. They're primarily F, H, and K, and C, very little of R1A and J2. There's very little IVC component in them. Todas, they're a combination of both the ancient layer and the Indus Valley layer. And Vadama, the Brahmin group, contains a blend of all three, R1A being the dominant one, but they also have the ancient layer of H, F, and R2. So it's also a blended culture. And then the dryland farmers, sort of the middle class folks, they're also a blended culture. One of the important messages is everyone in India is blended of all these three major layers, if you will. I included this slide primarily because there's a common belief that if you know your genetics, you know your mother tongue, if you will. That's not the case. The language you speak is probably related to where your ancestors lived the last few hundred years. And so, for example, this study shows the Jat community is focused on Jat community living across both Pakistan and India. And 1/3 of them belong to haplogroup L, which is an Indus Valley marker. But you also find this, and of course in South India, who are Tamil speakers. So the language they speak is classified as Indo-Aryan, and of course Peggy Morgan will say something else. But they, several thousand years ago, may have spoken a language more closely related to a language that is spoken by folks in Indus Valley or folks in southern part of India, if you will. So this is a sort of an indicator to point out that the genetic heritage and language you speak may have a weak correlation, not a strong correlation, something to keep at the back of our mind. Now, let me come to a very important study that was published about 2 months ago. This came out of Murjani's work. So her focus was, she looked at nearly 3,000 people in India. Unlike heritage, she looked at the whole genome of current day Indians, if you will. It's an important, so she's dealing with rich data, not sparse data. And she's raising many different questions in this paper, but I'm going to focus only on the heritage side of it. And by the way, this entire data of this 2,700 people or 3,000 people is available at her and via the website, via her supplemental data if you want to do any mathematical calculation. And where she sampled, the samples came from various different parts. She divided up the India as a north, south, east, west, and middle. And of course, the same color is consumed. I don't want to spend time on this. I'll go to this slide. This is an important slide. There's a lot of other data here, and I picked this up for the following reason. She takes 3 model groups. Sarasam. So Sarasam is in the Indus Valley margin, which primarily contains R2, J2, and L. There is no R1A there. So there is not ancestral North Indian. This is purely more representative of Indus Valley. And then she takes central step, middle and late Bronze Age, that's what MLA stands for, which is purely R1A genome. And then she takes Ongi, which is the Andaman Islands. They are AASI, ancient, the first Indians, if you will. Closest thing we have to first Indians. So there is no ANI, there's no ASI here. So these are pure genome that she takes. And then she looks at the different regions. And what do you need to, let me start with the south. So the green is, the label is here. Green is steppe pastoralists. These are the folks from Central Asia. And as you go from south to north, it increases. And the box statistically is the standard deviation, and the horizontal bar is the arithmetic average. And the dots are all the way through. And in the samples that she looked up, it could be as high as 30% of steppe ancestry, the whole genome. And it could be as low as 0, if you will. So that is the range, but the average is more like about 5%. That's going back to the left-hand side. This, by the way, the 0.2 is to be read as 20%, and this is 10%. And percent ancestry refers to autosomal DNA, that is the whole genome, not just a Y or a mitochondrial. Now, if you look at, so one trend you see is the amount of ancestral, the amount of steppe ancestry increases as you go from south to north. And I think many of us knew this, but this is a quantification of that phenomena, if you will. And now, if you take the blue, which is the Andamanese, the ancient Indians, the first Indians, they're pretty much somewhat, it's higher in the south, it's lower in the north, but the trend is it's present at levels of 30% or higher. And of course, it can be as high as about 70% in some communities, and it can be as low as 20%. It's never lower than 20%. That's an important point to remember. That means when you're looking at an Indian from somewhere in Bengal or in UP or Himachal Pradesh, he has 20% of genes from the Onge tribe, if you will. And in South India, he probably has a higher percent of it. Now, let's take the Indus Valley, which is the Sarsam as a proxy, which is labeled as orange color. You find that distributed, if you will. In South, it's probably as the same as the North. It may suggest, I'm guessing, there was a preferential migration towards South. Some people have argued it. That might have been the case. Maybe they migrated because of warmer weather, maybe because it was easier to grow things. But you find it to be lower in the East That was too far, perhaps. There were mountains in between to cross, rivers to cross. But you find in central to be higher is in reasonable number. So the averages here may point towards the preferential migration. Now, that is a hypothesis to be tested with some detailed calculations. Now, the nice thing about this particular result that she published about 2 months ago is this is using whole genome analysis. You're looking at the whole person, not just the heritage, which refers to where that person came from, not what he is today, right? So in that sense, in my books, this is a very important piece of result. Now you may ask, how did the people look like? Of course, all of us have our own visualization of. Now, from IVC, we have residues of the sculptures, and I've just taken what is available from the museums. And now that gives you an idea of how people looked in IVC. They were not homogeneous. They were highly variable. For example, the dancing lady had a jutting, thick lips, suggestive of negroidal features, and broad face. And of course, he also had, here is another sculpture here, which has more negroidal, more butted against. And of course, his nose is flat, just like the dancing lady. But he also had this gentleman who had more alpine features. Maybe he was of a J variety and more longish face, if you will. And here is another person with a broad and a longish face. They sported a beard. And here is a spear thrower who had a jutting out face. And of course, this skeleton was analyzed by Kenoy's lab and found it to be more African than typical Indian. He localized to East Africa, if you will, and that paper is available. So the people in IVC were highly varied, just like current day India is. They perhaps were a light complexioned and dark complexioned. They had thick hair and thin hair too. And they had thick lips and thin lips too. And they were quite a population, if you will. Now, if you want to know how ancient Indians who first came into India look like, probably the closest, she used Onge trap. These are pictures that I took from the tribal museum that's not very far from Ooty proper. It used to be near the botanical gardens. They moved it to a separate location, very nice location. It's a very nice facility. If you're visiting Ooty, please do drop in there. It's a very nice collection of the various communities that are present in Tamil Nadu and elsewhere. And this is how a person who lived in Madurai might have looked at 60,000 years ago. Now, that's a hypothesis, if you will. Something for you to take home, if you will. Now, one other implication of genetic analysis is we can find out how people migrated, not only based on autosomal DNA, which I shared, is the implication of one particular gene that's present in all of us, which is called as a lactase gene. LPH is how scientists call it. People who lived on milk and the products, this gene was modified when all of us are able to digest lactose, which is a sugar that's present in milk when you're young. When you grow older, that particular gene shuts down and you cannot digest lactose and you become lactose intolerant. If you're lactose intolerant, it gets digested by a bacteria in your GI tract and they produce a lot of gas. You have bloated stomach and of course a created GI tract problem. And so people avoid that. And of course, when you ferment it into yogurt, that lactose is converted and therefore there is nothing else that would be present. And maybe the presence of yogurt as being an important part of the diet could be due to people not being lactose tolerant. Anyway, what the chart shows, goes back to 2012, is a particular molecule in a location -13910. That's not important on a particular chromosome. When it changes from C to T, cytosine to, I think it is thymine or tyrosine, that lactose intolerance can just one molecule change, that mutation will make you lactose intolerant. The steppe people who came into India, they had herds with them to provide nutrients. They were not farmers. And therefore, they had adapted to living on milk, and they had this lactose tolerant gene, if you will. As they mated with people in India, that mutation passed on to their children, and people in the Northwest were more lactose tolerant than people in the East. People in Odisha, for example, they're lactose intolerant. Of course, the colored map is an estimation based on measurement made from those cross points. So this is a broad-based genetic analysis of Indian population. And so, and I'm sure all of us would know somebody in your family who is lactose intolerant, but not all of us are lactose intolerant, right? Because we have had that blending of genes, if you will. Okay, now let me bring all of this fancy talk down to earth. There are 3 of us in this group, and this is one of them. And so his genetic analysis tells us, and of course, this is a commercial enterprise that provides us data, and they go by they don't do ANI or ASI. They primarily do Indus Valley, and they define what they use, if you will, on the right-hand side. And so this particular individual has 40% of ancient ancestral South Indian. That is his genes, 40% of his genes are from the current day Onge tribe, if you will. And then Indus Valley, which is J, L, and R2, is about 30%. And of course, bacteria, BMAC may be related to Indus Valley, and therefore there's another 13%. And then he has a little bit of steppe genome, and of course, a little bit of other things included in it, which could be, sometimes these commercial enterprises could make errors too. But I just point out, predominantly ancestral, ancient South Indian and Indus Valley heritage is this individual, and he's one. So we should ask him to contact his great-great-grandfather as to what they spoke. This is case number 2, 50% AASI, the first Indians, and 20% Zagros Neolithic farmer. And the remaining part, I've boxed it into 50% core is ASI and Iranian farmer related as 22%. And steppe is 17% and Far East is 8%, if you will. So he has a higher steppe related one. Case number 2, both of them are very high on AASI, right? Here is a third one. He's primarily 40% AASI, and the remaining is substantially Indus Valley and AASI. Very little of steppe folks, only about 2.6%. So right here, 3 random sample, if you will, from our community have a blend of different proportions of these 3 major strands, right? AASI, Iranian farmer, and the steppe-related genome, right? So that's the genome part. Now I'm going to shift quickly to linguistic landscape. I'm going to try and do this briefly. How am I doing in time-wise?
Maybe you can continue, but it's 10:30 here.
So you're not going to mute me, right?
Never. Please continue, yeah.
So that's an authority you have, right? Okay. Now,
No, I just want Balaya to comment because he asked for 1.5 hours, man. He said he can be available for a max of 1.5 hours. Balaya, is it okay for you? You are on mute, yeah. You are on mute, yeah, Balaya.
I think another 10 minutes, 5 minutes, 10 minutes. So you can
I'll do 5 minutes and close up.
Yeah, absolutely. We are enjoying. Please, Mutharasan, you carry on. We are just listening.
Okay, fine. I'll finish this in 5 minutes.
Yeah, please.
This is a new area for me, and I think for most of us. And this area was added on, inspired by the book study. This book study group has been studying over a 2-month period the details that has been put forward by Peggy Mowen. The key thing to remember is a substantial number of linguists in this field have looked at primarily how words move from one language group to another language group, and they have looked at grammar too. They also have looked at phonemes, etc. But this person, Professor Mohan, has looked at, she puts forward the hypothesis that the foundation of a language, the skeleton of a language is its linguistic, is its grammatical makeup, which she calls as a skeleton. And the words in that language are really flesh. And the flesh can come and go, they heal and change with time, but the skeletons don't change or rarely change unless there's an implant or a surgical implant or something like that. And so that is her theory. And she argues right through her book, substantially in India, people speak a language that is very closely related to a very ancient language that was spoken maybe 6,000 years ago, maybe even earlier. And there may be substrata of different language groups that have laid over on top of it. And the bottom of it, I've showed here that Elam, which is southeastern Iran, she doesn't get there. That's my favorite topic. Indus Valley to Punjab to Gujarat, Maharashtra, Karnataka, and Tamil Nadu, the foundation of the languages spoken in that region is essentially the same. That's one of her results, if you will. And she makes example arguments in that. And the other part she also makes is the language from Indus Valley to Pashto in Pakistan to Burushaski, which is a language isolate spoken in Karakoram in the high mountains in the Himalayas, have a continuity too. And she also explores that. So I'm going to give you just a flavor of this. One of the things she focuses on is the retroflex phoneme, which is like ra, na, la, and ra, and such phonemes that is present commonly in Tamil. And also it's present in many other languages. And this is a map that she has used in one of her talks. This is a sort of a rough map. This has been studied by a lot of scholars in this field. And so she points out this continuity of this phoneme that is fundamental in many languages that is spoken in this region. You'll find a dense South India, retroflex phonemes, lighter as you go up north, and lighter in the Punjab region. And of course, it is less true in the eastern part of India, but it's present actually if you go up into the Himalayas, and I'll point this out. So she uses one of this as one of her arguments, if you will. Now, retroflex phoneme is important distinguishing feature, like ra in Tamil is rarely found in elsewhere. And apparently scholars say there is two versions of ra. I won't get there. It's a complicated subject. The other property she talks about is ergativity. This is quite foreign to Tamil speakers, but I will enlighten this. It is present according to her, would have been present in ancient Indus Valley. And this is, I think I have a separate slide that illustrates this. And the gender, for example, whether he, she, and it is what we have, avan, aval, adu. On the other hand, in other languages, languages such as Sanskrit, every noun, depending upon the ending phoneme, will have a male or female. A river would be a female, and a table might be some other gender, etc. So assigning gender to inanimate object is not part of the substratum in the ancient Indus Valley, according to her. And then the other is noun-centric. Dravidian languages are noun-centric. For example, even in Hindi, prem karna, love do. On the other hand, to love is a verb in English, and kadal panne, kalyanam panniko in Tamil, right? So marry is a verb in English, but in Tamil we say kalyanam panniko. Kalyanam is a noun, panniko is a verb. So it is a noun with a verb add becomes a sort of a special verb, specialized verb. And this is present in Tamil and all Dravidian languages, and also in many northern languages, including Punjabi, Hindi, and others too. So she points it out. Now, specific to retroflex phonemes, I've just listed here which is present where. And of course, I've compared only with what is present in Tamil and what is present in other languages. And interestingly, in Brushalski contains this rare phoneme of Ra. They have two different versions of Ra, according to scholars. One which is pronounced exactly the same way as we would pronounce in Tamil Nadu. They also have what is known as a slightly variant version. It's documented in the lower levels of the mountain. Higher levels, they still pronounce Ra, apparently. Now, they've, by the way, this is well documented in the research literature. There are 11 or 12 phonemes in retroflex phonemes based on how your tongue curls up. And 12 of them are present in Brushalski language. There are three major dialects. Some have 8, 9, some have 11, 12. And so there is a variation depending upon which part of that particular mountainous region people live. Brahui still has some of them, interestingly. On the other hand, adjoining areas do not have as many of them. Now, this is her table, Peggy Morgan's table. She looks at retroflexion as an important characteristic, and the other two phonemes she looks at are voiced aspirates and voiceless aspirates. Some of them are present in Tamil, some of them are not present in Tamil. And subject, object, verb order is right through true. I may point out here, the mother language of Sanskrit being Indo-European is subject, verb, object. I go to school, like in German. On the other hand, Sanskrit version is it an adopted feature or it's a native feature is something to be looked into, and that is an entirely different space anyway. The other feature that is present in the Dravidian languages and all the languages on the left-hand side is postposition. Wheat till send rain. Garme, right? You add the preposition as postposition. It comes after the object, right? So that is present in Hindi, in Punjabi, and of course in Tamil, in all languages, in substantially in language. It's absent in Sanskrit, if you will. Light verbs, where you have gender, I think Venture Ergativity, I have a separate slide. Compound verbs, where you can stack up multiple verbs together for you to express a particular expression in Tamil, for example. I remember somebody saying you can stack up as many as 8 verbs, one after the other, without a conjunction. And then conjunctive participle, you have part of a verb combined with another verb in order to express a particular view. Sendru Vandan, 2 verbs together. Sendru is part of a verb, not a full verb. And Vandan is a full verb, so that is another way of communicating. Reduplication, you know what you mean. Sala salanu, satam ketudenu. So the repeat of those words. And then echo words are in a similar way. And honorifics, I think that's something that you might have heard about. I just want to illustrate. So she compares these features as language X. How many features are not present in current? In Brushalski 1, so she says that might be the closest language to language spoken in Indus Valley. Brahui is the second closest, and Punjabi and Tamil are the third closest, according to her. This is from her book. And Rig Vedic Sanskrit is the farthest because there are 7 features that are absent in Sanskrit that are present in her suggestion of language X. So the natural question that comes to me is, did Tamil have ergativity features in ancient times but lost it to be examined? Is it present in tribal languages? I've read some literature suggesting Toda language has split ergativity. Maybe it is still present. That's something to be examined. I'm not sure people have examined it as yet. Now, this is the illustration of ergativity. For most of us who are Tamil speakers, this is a foreign concept. Here, if you're a Hindi speaker, this is something you'll be familiar with. The noun, when you're using a transitive verb, that's a doing verb, the noun gets modified in past tense. But so ergativity comes in different versions of it, full and split. And in Sanskrit, it is absent. It is written up here. I'll look at only the transitive verb where there is no modification to sita, you see there. Same in Tamil, there is no modification to sita for that sita sapital. And same in Brohi, there is no modification. There's no, it's a simple accusative form. While in Hindi, it'll be a modification of ne, sita ne kaya. So the subject in past tense becomes ergative. The subject gets modified when you're dealing with a transitive verb, that is a verb that does an action, if you will. And same thing is Burukowski, sita ne kui, which will be the subject gets modified. Her thesis is that ergativity was part of the property of Indus Valley civilization language. Languages do change, as all of us know. They do evolve, and some of the features may get dropped out or maybe added on, and so something to be kept in mind. So this is my last slide summary. I think I gave you a little foundation on heritage, mitochondrial DNA, and Y haplogroup. And then I told you a little bit of how this field of ancient DNA evolved starting in the 1980s. And then the two major groups, Stanford group and the Harvard group, and of course the Swedish group, have contributed a great deal to this field. And of course, our Indian scientists, Dr. Tangaraj and Professor Pichep. And then I shared with you the 4 ancestral strata of pretty much every Indian. Doesn't cover everybody. I'm sure there are other people who came into India, but their primary genetic strata. And then told you how the chronology was determined using genetic admixture model, how that came about. So we have a better picture of this only in the last 10 years. We didn't know this before that. And then we gave you three examples of people in this community, and many of them fit into this model of this ancient ancestor. And then I gave you an idea of language X hypothesis of Peggy Morgan and some structural features, right? I'll stop there and answer questions. Oh, suggested future work. A professor never stops without finishing up without a question. So the phonetic characterization of tribal languages, this stems from my own experience, direct experience speaking with Toda people. I went to school in Nilgiri district, Ooty, and so I've interacted with Todas. They have a lot of aspirated sounds, if you will, and of course these are absent in Tamil. Is it possible some of the tribal languages harbor some of these phonemes that are now absent in Tamil, and that is suggested to be hypothesized to be present in IVC? Second thing is, I've seen a lot of studies of tribal languages by themselves, but I haven't seen, maybe it's available, comparative study of tribal languages, just like how Dravidian comparative languages have been done by Amino and other scholars and their followers such as Krishnamurti and Deshpande and several others. Similar studies of tribal languages might shed some interesting insight. And then ancient DNA characterization of excavated skeletal remains from India. That work requires. The problem there is the fragments we get are small, not big. Typically, the ones that you find in cold climates are about 1,000 nucleotides. What you find in hundreds are a factor of 10 less. And therefore, the puzzle is it's geometrically very large puzzle to be assembled. I think this will be solved, in my opinion. It's a question of people putting their brain to it. I'm particularly alerting the data scientists in this field because it requires a tremendous amount of computational capability to do this type of work. In fact, that's a field in which Professor Wright got into after his PhD, and that is why he's been able to make major progress in this field, by the way. Something for the IT folks in this field who work in the data intense field would like to get into. The final one is the most recent paper from Murjani's article is, we know at least 162 Tamils whole genome, autosomal genomes are available, and it's available to download and do calculations. And she hasn't done that. She's done that with respect to broad population, but the detailed analysis of this could be done. That could be a nice research paper in a major research journal, something to look at too. And now my basic question is, if tribal languages harbor some of the ancient features of languages we now speak, is it possible the language you speak is the elite version of ancient language that was present in India long time ago? Okay, I'll stop there and open up to answer questions. Or
Thank you so much, Dr. Raj Mutharasan, sir. Excellent presentation. Heavy subject, but you rocked it because with your clear slides and easy explanation that anybody could understand. And I personally felt your calm, even toned voice was a big plus for this presentation, because the listener, as myself, we couldn't get distracted. We were constantly being drawn into your talk. Thank you so much.
தூங்கலன்னு சொல்றீங்களா?
யாரும் தூங்கலைங்க, யாரும் தூங்கல. ஆக்சுவலா மனசும் மைண்டும் ரொம்ப விழிப்புணர்வா இருக்குது இப்ப. ரொம்ப தேங்க்யூ. It was a very enlightening speech too. Thank you very much. And I call upon Mr. Mani Manivannan to hold the question and answer session. Mani Ayya.
மணி ஐயா பாலாயாட்ட கேட்டுக்குவோம் உங்களுக்கு.
ஆமா
ஏன்னா
ஆமா அதான் நல்லது.
10:45 ஆயிடுச்சு. நீங்க மியூட் ஆயிருக்கீங்க பாலாயா.
Bama, I have no problem.
Question answer முடிச்சுட்டே பண்ணிடலாம்.
Yeah, absolutely.
Okay, சரிங்க.
Oh, okay. Thank you, sir.
நன்றிங்க ஐயா.
Thank you, Professor. Like Subhatra said, it didn't feel like a boring seminar on a lazy Friday afternoon in graduate school, but it was a very exciting first presentation on a Monday morning, 8 a.m. So it was very good. I enjoyed it. Now let's open it up to questions, and I know that as a professor, you would enjoy interacting with the students. So any questions from the audience? I don't see. You may want to turn off the presentation so that we can see everybody else. Okay, go ahead. Any questions from people?
I have one. So,
okay, please go ahead, Bama.
Yeah, in that 2019 paper from Narasimhan, which talks about the genome, like he studied the genome population and genome, he made the genome studies and made a detailed study on the endogamy, right? That and this Murjani's paper, the recent paper, what information in Murjani's paper is adding? I am not able to visualize, I am not able to understand Murjani's paper in a better sense. Like you said, it is more of the genome studies, but the bar chart, whatever you provided, the box chart, whatever you provided, it was more about the heritage, the genetic heritage, right? So what, because it is a complete genome analysis, we can have more information from that. So
to answer your question, there are 2019, there are two papers with Narasimhan, and the first paper is Narasimhan, there is no Shinde there. Second paper, Shinde first author, second author is Narasimhan. The major difference is the following between the Shinde paper includes the Rakhi Gargi. The earlier paper does not include the Rakhi Gargi lady. The earlier paper looks at the whole generation, and they hadn't yet gotten all of the data of ancient DNA. So at that time, he still speculates how the steppe people came into India. But in 2019, that is clearer. The fact, the trajectory of the steppe folks was via Hindu Kush region. That distinction really evolves around 2019. That was a major contribution. Although the paper talks about Rakhi Gargi lady, that is what I got as an important contribution to it. Okay. The second important one in the second paper, where Shinde is the first author, is that lady harbors a substantial genomic content of the Andamanese people and the Iranian hunter-gatherer, not Iranian farmer, right? That's an important distinction.
Hmm.
Did I answer your question?
Mutharasan also, if I can say, that Vasant Sinde's paper also spoke about the absence of steppe ancestry.
Yeah, it did.
So that is an important point. That was followed by a press conference next day. So that is more important. That
Where he
means,
contradicts himself.
yeah, because he was the primary author in the Cell magazine, a scientific paper, followed by a press conference next morning, which was sounding something different. So that is where the politics of the history emerged so evidently. Probably we will discuss that. So more important, the Rakigadi output from the Sinde's article is absence of no evidence for the steppe ancestry in that particular ancient DNA.
That is correct. That's absolutely correct. Of course, the public, the media spun it in very many different directions, unfortunately.
Yeah. Because the headlines was more on the base of the press conference, which he was asked to do, rather than by the scientific paper, which he wrote.
It's unfortunate. In fact, he in fact says, and I listened to that press conference.
I do listen.
He said she was 6 feet tall, just like any Punjabi lady. I mean, bizarre. She was 5'4". She was a tiny lady, small, diminutive proportion. Anyway,
In fact,
but
it was a negotiated settlement. Even the terminologies like how Vagis Narasimhan and Vasant Sinde is negotiated for the AASI, ASI, ANI, all these terminologies were the negotiated terminologies. To then the scientists still stood to their guns, saying that there has to be fine difference between ASI, what they chose to call AASI. So that's what they try to put their scientific storm. But at the end of the day, if you have to do an ancient DNA study, you need the blessings of Vasant Sinde.
okay, that's a different, separate topic. And then let's get down to questions.
Sir, I would like to say something about it. First, it was a phenomenal, absolutely stupendous presentation, as Subhadra Madam pointed out. I am only coming back to what I spoke to you over phone some time back. I know that you are a chemical engineer and then with so much of scholarship in engineering and science. But then this is the domain which we need basically a book from you in a very lucid way. And then if it requires a kind of a manpower support, the Indus Research Center of the Rojamutia Research Library, where you delivered a talk, we would be in a position to put a person at your command. And it is a time that somebody is putting a transdisciplinary perspective with a scientific rigor, at the same time in a lucid way, and the way you put it, a multi-spectral, like ancient DNA and then linguistics data and so many things, cost. So I take a little liberty of taking some time with you. I would like to relate to some of the topics which you touched. So let me start with the lactose intolerance. I have the experience of meeting, having worked with the tribals of Orissa, who speak 62 types of languages, distinct languages, belong to Indo-Austroasiatic and then Dravidian and other subgroups. So then I visited a tribal village in 1988 to investigate whether the animals, the cows and buffaloes, distribute to the tribals for the purpose of them to sell the milk, whether those buffaloes are still alive. Whether it was really given or somebody made a corruption. I walked kilometers after kilometers hills and then went and met them. Most of them had eaten that buffalo, not milking. So when I inquired about it, they asked me a question back: will you sell your mother's milk? Then I was taken aback by this question because I came to know that tribals had never milked an animal. They never had drank a glass of milk ever, and it is beyond their culture. So I investigated the tribal groups of Orissa into two categories: the tribals who take the milk to tribals who do not take milk. If you really look at it, Toda population, in spite of its ancientness, revolve around the dairy and the buffalo milk and other thing, whereas the tribals in the eastern India, they are absolutely not drinking. Probably if you study them scientifically, they will have a lacto intolerance. So that's, as you rightly pointed out, Eastern Indian tribes, I have the personal knowledge about it. Number two, I will come to that why there is a dearth of ancient DNA material or the kind of evidence available from South India. I want to bring to your notice that when Satyamurthy came back from Orissa and started digging Adichanallur after a gap of 100 years after Alexander Ree, he started doing it in 2004. And then they came across a full skeleton. And then Satyamurthy, being Satyamurthy, he informed Anthropological Survey of India and the ASI in Mysore. The scientist came and carefully, the anthropology people, they took it for the further test, and no one know what happened. In my book, I mentioned a separate paragraph called the skeleton in the Adichanallur cupboard. So skeleton in the Adichanallur cupboard, which I have a title, because neither Satyamurthy know it. We asked the question under the RTI. We asked the people to find out what happened to the skeleton. And no one has got an answer. So probably people would not like to have, of course, when Pichappan did a study, and Pichappan's further study also, I was involved also. Some of the articles contains my name also, though I have not contributed, except the selecting the villages for the DNA studies. So that's what about the ancient DNA, the dearth of it. And then, of course, I would like to say you connected on one side a DNA material haplogroup, and then from the historical data, how the data is functioning. On other side, the end of the thing, you connected that with the linguistic data, retroflexion and other thing. So then everybody knows that there is a Dravidian substratum. Not only Dravidian substratum, there is an amount of Austroasiatic substratum also. Some people call it a language X, and particularly the syntax is a very important feature of any language, like a sentence SOV. We said that particularly the retroflex, many things which Peggy Mons also pointed out, you also mentioned about it. But I would like to say, without having access to any of the DNA data, without having access to the Peggy Morgan, Tony Joseph, and the Vagish Narasimhan, Vasant Sindes of the day, way back in 90s and 2000s and 2010, we started talking about what is called KVT complex, Korkai Vanji Tundi complex, the place name complex. I have an offer. The data you have based on the DNA, how it works out, different communities, different locations, different geographies. If you superimpose that data, I have a few offers. If you superimpose on what I suggest as a toponymic data of the KVT, Korkai Venji Tundi complex, and the RIG complex, Rig Indra Gandhara complex, which last year I gave a talk, which right now I am writing a book, The Horse Movement. If you superimpose this data, I am pretty sure, number one, place name data. Number two, I have another offer suggestion. Like in India, I made in my book, talked about the what is called pot root, like silk root and spice root. I spoke about Panai Tadam, pot root. So then many people miss that bus. How the painted graveware, that what you spoke, recent DNA is telling, rather than in Iranian and that hunter food gatherer farmer movement, there is a movement from the Hindu Kush side. So that was exactly get into the upper Gangetic Valley through the Gangotri, Yamunotri side and up to the Haridwar side. This is called upper Gangetic Valley. This painted graveware forms an island. I have put a map in my book, Journey of Civilization, is the painted graveware. And then I am claiming the block and redware, which is done through the inverted firing. And that is the pan-Indian pottery, which I put it in the map, how it is superseding, overlapping with the PGW area in the eastern side. It is overlapping with the northern block painted ware in the eastern side. And then completely the absence of painted graveware in the entire South India and presence of the northern block painted ware in the Adichanallur, Archimedu type of places. So if we put the data put together and if you superimpose DNA study, the linguistic study, it will match. And I think if you put your data on the high west, low east hypothesis, which I have been talking about it for years together. So what I'm trying to say, what this modern science again and again proving on the basis of the deciphering the body language of the, I still batting for the, one is the DNA data, another is the linguistic substratum and superstratum, and the third one is the undeniable cultural markers, which you cannot change. Nobody can deny that there is an absence of horse in the Indus Valley, the entire inscriptions and the field symbols. So there is no horse. So Rajarams, they tried to invent in 1999. So now even everybody knows that there is a horse arrived subsequently. If you study the horse, if you study the arrival of the chariot, then how the South India horse arrived through what route, then you will find the answer for the many of the questions which you have raised. That means that as you rightly said, there are a lot of people in India, particularly in Tamil Nadu, believe that caste means language. Actually, caste does not, even the language and the DNA is not exactly equal to each other. There is a huge amount of overlapping. Forget about the caste. That here the people, guys are talking about, you are a Tamil caste, you are a non-Tamil caste. I have been telling them that the secret known only to the grandmother is the name of the grandfather. That is what the science is again and again telling. Again and again telling there is an incipient mitochondrial DNA and incoming Y chromosome. This has been the history of the India. That is the reason I have been talking. India is not a melting pot. India is not a salad bowl. India is a rainforest. So that is the ideology and the cultural politics which we have been trying to make sense in this country. And the Dravidian substratum and the Indo-Aryan superstratum and the arrival of the horse is a dialogue between the horse and the buffalo nation and the presence of buffalo and many markers. That is the reason I've been telling the deciphering. You may not be able to decipher the Indus script, which the Tamil Nadu Chief Minister has announced $1 million. But whether you are able to decipher or not, the DNA will speak truth. More importantly, we are deciphering the body language of the different parts of the. There is absolutely no, like a Troutsman studying about the Dravidian kinship marriage system, cross cousin marriage, and the parallel cousin marriage, and then how the Magatpar Kanji type of thing comes in the Sangam literature. So it's you have opened a kind of a transdisciplinary window. My request only, you must convert. I'm sorry to say that you must. You should convert this into a book, and the Indus Research Center, which I am heading, which is a part of the Roja Muthaya Research Library, now is going to be Tamil Knowledge Campus, willing to put a qualified person under your command to help you and co-work with you.
That's a lot of promise. I will put it together. I will put it together. I may need help with graphics, but text I can put it together.
Wonderful.
Thanks for your comments.
Thanks for your comments,
Thank you, Mani.
Balakrishnan. There are at least 3 questions that I see. Sridharan Jagadishan, sir, please unmute yourself and ask your question.
My question to Raj was about the slide I saw on, it's called Chennur 8,000 to 40 years, which is a surprise. So can you please elaborate a little bit on that, sir?
What was it? Chennai?
Chennanur. He's talking about
Chennanur.
Chennanur
Yeah.
result.
Chennanur is near Tiruvannamalai and
Krishnagiri district. Krishnagiri district.
Krishnagiri district. And this came out of archaeological report that was published by Tamil Nadu Archaeology Department sometime about maybe 3, 4, 5 months ago. And what they discovered was they were able to go to the virgin. Typically, archaeologists, what they do is they start digging and they characterize each layer, and they went all the way down to what they would call as a native layer. That is, there's no residue of any human habitation. That layer, just above that, they found pottery, and the pottery was OSL dated to that particular date. My reservation on OSL is it's got a lot of errors, but it's not error in thousands. It'll be errors in the orders of hundreds. So they have a hard datable object of 8,400 BC. That's about 10,000. And Mehrgarh in Indus Valley was supposed to have been settled around 8,000 BC, which is almost a contemporary of Mehrgarh. Mehrgarh is considered the first settlement to have started appearing in the Indus Valley. And of course, that's where buffalo was domesticated. And they had domesticated cotton there too, because fabric of cotton was discovered there. And there were skeletons that were found. Interestingly, I think, was it there? Trying to remember. They actually buried their dead in their living quarters. That is when a person passed on, they'll bury it in their own home. So they must have believed the dead were part of their family, even though they were dead at that time. And so it's worthwhile looking into the literature of Mehrgarh. I'm trying to remember now what I had read maybe 10, 15 years ago. There's a considerable amount of work has been done on that, and it's well documented because Kenoy was involved in this excavation, and it's very well written up. So that's something to look into. But this place in Chenanur is more recent. I'm sure they're continuing with their excavation, but we'll have to see what evolves over the next couple of years.
Thank you, sir. Thank you so much.
Let's go on to Chundarachoran. Sir, please unmute yourself and ask your question.
Hello.
Yes.
சொல்லுங்க.
Yeah. It was a very excellent presentation. I enjoyed it very much, very informative. I've got a lot to digest. Thank you for that. I have only 3 questions. One is, in one of your slides, you mentioned about the migration of Iranian farmers into the Indian subcontinent. Initially, there was some opinion given about hunter-gatherers migrating into India, and subsequently, probably I got a clarification that it is only Iranian farmers who migrated into the Indian subcontinent. Now, my question is, how do we differentiate between people who migrated, whether they were hunter-gatherers or farmers, whether we have any specific DNA markers, on what basis, on what kind of scientific analysis we are able to differentiate that. So that I would like to understand. Probably there was some confusion in the initial stages. Subsequently, it was clarified. I understand. Point number 1. Point number 2.
No, before you go to the second one, let me answer the first question.
Okay.
Then I'll forget your first question if you want. Okay. The basic tenet of this is, for example, it's not uncommon to see conflicting interpretation. The key thing that all of us must remember is data, and interpretation of the data could be different depending upon what new knowledge and new information comes to the fore. For example, in the paper of Raki Gargiledi, the genome was based upon from Zagros region and Andamanese Island. The Zagros region DNA was associated with pre-farming, and therefore they interpreted her as a person of formation of hunter-gatherers and didn't have an Iranian heritage, if you will, Iranian farmer heritage. Now, on the other hand, when you look at the people who moved into Indus Valley, you find L, you find J, and R2. J particularly is associated with farming, and therefore that has been interpreted by scholars. Now we are looking at a different time frame 5 years later, because that publication is 2024. They have interpreted that as Iranian farmers moving into India. That is how expansion of haplogroup L1 is explained. Okay. So do you see the distinction between these two approaches? So both scholars, they're all both distinguished scholars in their own field. They're interpreting the data in a different way. So the way to read the Rakhi Gargi paper is, her heritage was the genome of Iranian hunter-gatherers. On the other hand, the later paper shows farmers in Zagros region as being the people who moved into Indus Valley, who are now currently in Tamil Nadu, Singapore, Sri Lanka, etc. Right? You see the difference? Now, it's not uncommon in an evolving field in science for people to have conflicting interpretation with the same data. But as time goes on, they'll get settled into some constant value. And I used to cite this as an example in previous talk. We thought F equal to ma is universal truth from 1500s all the way till 1908, until Einstein said that's only an approximation. It requires at the bottom square root of 1 minus velocity divided by velocity of light squared, square root sign, right? So the F equal to ma was modified into a more refined equation. So which is true? Both are true, and the truth has a particular time, right? So we have to keep that at the back of our mind. This is an evolving field. Let's keep that in mind. Remember, we're also not dealing with a lot of ancient DNA from India. So we are trying to estimate many of these things using what is surrounding us. So one of the fundamental principles all of the scholars field is what they call as a genetic gradient. That is, if I were to take a population in, say, in Chennai, take a population in, say, Virupuram, take a population in Madurai, and if I were to get a statistically representative sample, there would be a genetic correlation among them. They won't be very different. There may be one or two people very different genetic heritage, but there will be a continuity. Similarly, linguists also make the argument as linguistic continuity. So the gradient is what they rely on to predict what was there in IVC using information of genetics in the surrounding area of Indus Valley, because we have only one representative from Indus Valley, right? So keep that as the back of our mind. As more skeleton gets categorized, these things may get refined to a better form, and more closer to truth will be found. I can tell you 15 years ago, we had no idea how the people in Orissa spoke a language which had no relationship to people who spoke in not just a few hundred kilometers away. We knew they had a different language. You know, they were all men who came from current day Myanmar and Thailand, but they married Indian women, so they had Indian features. They didn't have the so-called Eastern features. But on the other hand, when you look at the genetics, you know where they came from. We are able to link the language they speak and their genetics very beautifully. And now we can tell when they came. We did not know that 15 years ago. Now we know when they came. Second question.
Okay.
That
Before you do
naturally.
that, okay, just a minute. I have to sign off because the internet technician is here. So, Bama, can you take over from me?
Yes, sir. Yeah, I will take care.
Okay, thank
Thank
you.
you very much.
இத்தனை முறை இவ்வளவு நேரம் எந்த இன்டர்நெட் உபயோகப்படுத்துனீங்க?
மொபைல் சேட்டலைட் தான் வச்சிருப்பான்னு நினைக்கிறேன்.
ஸ்பெஷல் சேட்டலைட் அப்பா மணி?
This is
ஐடி ஃபீல்ட்ல இருக்காரு, அதனால ஏதாவது ரிக் பண்ணி வச்சிருப்பாரு.
இல்ல it's there is unstable connection, so
I see.
AT&T has to come and fix it. So periodically I lose you, but fortunately they didn't cut me off. Good. Okay.
Unplug and plugging back fixes many of the problems, Aliyah. It's a standard engineering solution.
Power down, power up. Yes.
Okay.
So can I continue?
Yeah,
Yes, please.
Okay. The second question, your reply to the first question naturally leads to my second question. Um, about again the Iranian farmers moving into Indus Valley civilization areas. Now the question I have is the Indus Valley people, did they learn and practice agriculture on their own, or did they learn it from the Iranian farmers only? We have plowed field also, if I am right, it is from Kalibangan. So
That's
period
correct.
wise, chronologically, how does it, how do we situate it? Did they develop it indigenously, or did they learn it from the Iranian farmers? Because Iranian farmers means probably they could have learned it from them. What is the reply we have, sir?
The short answer is I do not know. The reason is that is an interpretation of existing data. What we do know
Okay,
is the following. What we do know is the following. 8,000 years ago or 9,000 years ago, people settled at Mergard. If you're settling, you're farming. People do, people who do not know farming don't settle. They go around trying to collect things. So,
correct.
and similarly, we know in 4,000 years ago, there were settlements in the Deccan. And now the Chenanur settlement of in 10,000 years ago tells us they were farming there. There were settlements there. Settlements there means they were farming or they were able to harvest enough food in the surrounding area. Now, so the hard answer is that would be ambiguous. The settlement at Merga was fairly large, and therefore farming would have been there. And when was that? That is about 8,000 years ago, 9,000 years ago. And when did farming begin? Around that same time in the Fertile Crescent. So they might have been contemporaries, or there might have been one guy from far away walked in there and said, "Why are you guys running around hunting? Let's settle down and farm," right? So there might have been something like that. Or so the hard answer for your question would be very difficult to get.
But
Now,
but
Kalimantan, you're now talking about several thousand years later. That's a different story altogether. That's a
yeah.
different time domain.
Yeah. Yes,
Understood.
Mutharasan sir, I would like to add here. When you talk about Megargar settlement, as you rightly pointed out, 8,000 years of a continuous settlement, in the multi-layered settlement, there is a unbreaking, and at some point of time, people started living in a proper houses, and also the first evidence for what I have been talking about, the dichotomous settlement, like say high west, low east kind of thing. That been what subsequently Tamil literature, Sangam literature, after so many after few thousands or many thousand years after that, it talks about the Irubal Peiriye Urgalu Mudur, and then the dichotomous village as a concept. The earliest evidence available in the Megarkar, notwithstanding your evidence for the settlement pattern in South India being available, the Megarkar is a gradual how a kind of a settle an agricultural village, get into the house, then getting into a small settlement, divided into two parts, then and then which becomes a prototype for the later on Indus cities. That's what we've been.
Thank
There
you.
is a very good of institute development there.
உங்க
So,
மூணாவது கேள்வி.
okay. Moonavathu. In one of your slides, you mentioned about the Koraga tribe. That's a recent research we have come across in the popular magazines. I have read about that. And what is the significance of that finding is what I am very much interested. How different is that from our earlier understanding of the populating of India based on what we have already? In one of the popular this thing which appeared in Indian Express, there was a mention about a correlation between linguistic and genetic lineages that is being established by research into this Koraga tribe. So that is more perplexing or more intriguing or interesting. So probably what do you like to add more about that? And it also says that there is an unknown strain of connection between IVC, which that is being displayed in Koraga tribe. I don't know. I just want to know some idea about that. Probably you can throw some light on that. It will be very helpful. Thank you very much.
Okay, that is a work that came out of one of the universities in Karnataka on the western end. The primary discovery that was made by that author was the Koraga tribe had a high presence of mitochondrial haplogroup U1. U1 is hardly found anywhere in India, as far as I know. That's the only tribe that has that. Now, the origin of U1 is now has been related to somewhere in Iran. And because there have been excavation that showed that mitochondrial U1 has been associated with that particular culture. So the author, in a very optimistic way, believed, I think it is purely his belief, said Dravidian language was born in that spot. That was a little too far-stretched interpretation in my view. But the fact that the other part that is interesting from my point of view is I looked into, after seeing that paper, I've read that paper, I've glanced at that paper, looked at Koraga language. Linguistic Society of India published the compendium on how the language is spoken. Amino in 1971 writes that Koraga language should be classified under Northern along with Brahui, because he finds certain characteristics in Koraga language very similar to what is found in Brahui. So that connection was very interesting for me, because that thought that crept up in my mind that the tribal languages are important storehouses of ancient Indian linguistic features. And my belief is they haven't been studied extensively and they are worthy. That's why my last slide had that particular bullet point that we really need to study them very carefully, fully. And so that is the connection. The fact that Yuan was found in Iran, Yuan was found in Koraga, and that there were speakers, a connection along with the Emino's opinion that it should be classified under, he said should perhaps be classified under Northern along with Brahui, makes that connection, right? That's a connection he also makes in that paper. But languages evolve and languages change. Making linguistic arguments fundamentally related is not strong. You need supportive other arguments to go with it. I'll stop there.
Thank you.
Thank you very much. But the only thing that was intriguing was the article mentioned about correlation between linguistic and genetic lineages. But this was not supposed to be the first instance, historical instance. That was intriguing. Thank you. I'll stop at that. Thank you.
Thank you.
Yeah, my question is like you talked about the J2 haplogroup migrations, right? When you talked about the tribal population in Tamil Nadu, they had a pretty much the significant composition of J2 haplogroup. But usually when we talk about the Iranian farmers or the IVC migration, we associate with the haplogroup L. So this J2 is part of the Iranian migration or something different?
It is, that could be confusing. I highlighted that paper on L2 because, L1, because they had done the Bayesian model on the evolution of L1 and how that connects to India, right? And that was done just about a year and a half ago. Now, J2 is found in the Zagros Mountains. Zagros Mountains contains J2, L, was it classified L1, I think, and also R2. And that connection is the one that connects to the Indus Valley civilization, if you will. And you find all three, and of course, one of our listeners is of that J2, J-L26, I think, which is the way scientists label it, but the commercial will call it as J1 or something like that. So it is omnipresent in South India. It is present in bulk of India, but not at very high proportion, but at about 5-10% level. Same as L, same proportion, and R2, same proportion. And those three, in my opinion, that now I'm getting into the opinion domain, C, F, H, combined with these three, might have formed the foundation of Indus Valley civilization, because there were people living in Indus Valley before the so-called Iranian farmers came, and they were doing other things there. So it would have been a mixture of that blend of people. So you see that in the Indus Valley statues, you find people with very negroidal features versus very alpine features. So it is clearly a multicultural society, just like how any Indian city is now, right? So that feature was present at that time.
I have two more questions, but is it right time to ask, or we can continue in the WhatsApp group? I'm checking with you, Aya, because you look tired.
Oh, no.
Okay. So the next question for me is like in that 3 case studies you presented, right? There was the case 3 had something called copper age Zagros. What is that?
Okay. The people in this field look at settlements as pre-metal age and post-metal age. So the copper age is one of the early ones, bronze age as they begin to alloy and figure out how to bend, and then comes the iron age. And of course, Tamil Nadu stands differently in this category. The evolution of civilization goes from stone to metal. The metal, they start with the copper. The reason is copper is easy. You can take a copper and bend it with your hand, right? And it also requires a lower temperature for you to extract from copper oxide. Tin is even liquidy, right? And of course, and they figured out at some point in time, when you put them together, you get a stronger material, bronze. And of course, somehow zinc entered in there, giving you even strength. And so that became an important. Copper age is one early stage, bronze age is a more developed stage. And then, of course, is the iron age. This metal stage goes through that phase. Before that, they were relying totally on stone and mud.
Yeah, my question is why? Because that classification also has Indus Valley civilization, which is also copper age, right? So
Right.
there is then it is distinguished separately Indus Valley civilization and then a copper age. Oh, this is much earlier. The time
Yeah,
period.
but copper age cannot be confined only to the Indus Valley. It's a continuous, this geography and the technological changes is continuously evolving. It happens in BMAC, it happens in the Jagros. So it's like a metal age, pre-metal age, certainly pre-iron. There was a copper age in that area and pre-iron, and then iron comes.
Okay.
I don't know.
Okay, so maybe this is the last question. So when you talked about the mitochondrial DNA, most of the Indian women has M, and one of the chart you showed the
Men
iron.
also have the same, not just
Yeah, sorry. All the mitochondrial DNA of Indians. Okay. So the Iyer and Iyengar also had 50 percentage of the mitochondrial DNA. But the skin color is, I think the skin color is the autosome has to do with the skin color. Um, like it's because the colors are different, right? If it is Brahmins or any upper caste, they are more fair. And in
Not
other regions also we see it, but it's predominant over there, right? So what brings the color, skin color? How do we visualize that in the genomic point of view?
It's a more complex question. The complexion is attributed to a particular gene. I don't remember the exact name. By the way, that work has been done. It's available in the literature. So that gene, just like lactase gene, LCT, opens up. High pigmentation occurs if you live in the sun. It's an adaptive response of your body. It's the biology. But what biology tells you, if you're always in the sun, you start creating that pigmentation, so you become darker. For example, Australians are fair skinned now on an average. 5,000 years after this, they'll be dark skinned like many of us here. So that's an adaptive response. So the people who came they came 4,000 years ago, they came from a cold climate. For them, the deep pigmentation was not there. As they came into India, that gene transferred as they blended, and so instead of biology taking over your skin color, blending muted that particular biological response, and some where they expressed that particular gene became lighter skin color, if you will. So that's a sort of a rough and tumble explanation, but there's a more detailed explanation that's available in the literature. This has been studied biologically, if you will.
Thank you. Okay, let's get to a close. Balayam, do you want to add?
Mutharasan, you mentioned about the JOT, the population. Particularly, I have traveled extensively in the JOT region, particularly in Haryana and Western Uttar Pradesh, that the entire JOT belt. If you, the DNA thing, it correlates in one particular way. But then a person like me always look at the what I call as a body language or a cultural elements which are little dominant. It does not mean that it is articulated only on particular specific area, but in a broader way. If I can afford to say that in the northern Tamil Nadu, particularly where predominantly the one-year community lives. And then the people who have made a study, you will be knowing better. The one particular community in Tamil Nadu, that way in South India, if it is in genetically which is more closer in a North Indian population, that tallies with the JOT population and the one-year population. But then culturally expressing that, I would somewhere talk about the Vanni. That's the reason in my book I am talking about the Vanni roots, where it is called in the Punjab and Haryana area, it's called Kejri tree. And then in other North Indian area, it's called Shammi. And then it's called Bunny in Karnataka, and then in Vanni in Tamil Nadu. Again, the state tree of the Rajasthan, the national tree of the UAE, and also the Mysore Udayars, and in the royal tree of the Cholas. And again, and the habit of Agni Sachi, the concept of when it comes to the Vedic rituals, even today the Vanni stick is used to ignite the first fire. So basically the Vanni Sachi becomes the Agni Sachi. If you go to the Sanskrit root for the Vahni, 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 it's basically considered to be the root word for the fire. So this Vanni is used for the igniting fire. And again, I would like to say in the Mahabharata story, when it comes to say 1500 BC, people are talking about a Mahabharata age, where it's again Vanni tree is correlated with the Pandavas hiding their weapons. Then again, the Draupadi cult, if it is somewhere in Tamil Nadu, if it is well pronounced, very dominantly pronounced, it's in northern Tamil Nadu, which again goes back to the JOT kind of a thing. So all put together, I am not saying that it's an accurate science, but then I make an argument that it is like a broadly makes a marker where to look for what.
Yeah, but it's a multidimensional issue. For example, genetics is one, linguistic is another, archaeological artifacts, cultural practice, for example, the way we speak, etc. There are many dimensions to this, right?
Marabu Kadagal and even folklore, carried forward memories.
Like I think I remember making this question. We have exclusive V and inclusive V, Nangal, Yengal, right? Nam Nangal. And you can trace it all the way to Rajasthan, that particular cultural feature.
Exactly.
All the way. And on the other hand, English does not have this cultural feature at all. There is no inclusive V, exclusive V distinction. There's only we, that's it. You have to have a phrase to incorporate that distinction. Anna, Tamil, you can just with one word you can express that.
Thank you all for the questions. And thank you, Mutharasan sir, again for being patient and clearly explaining all the questions. Really, kudos to you. You have done absolutely a wonderful job. Thank you so much. We all appreciate it. Thank you. And now we are coming very close to the final ends of the session. I'm going to call upon Mr. Balakrishnan, IAS, who has been here, who has been interacting with us, to give the final concluding remarks for the presentation today. For all the viewers here, it is he who is the main reason why we have all been clubbed together as a group. Thanks to him and his lovely book, The Journey of a Civilization. And that's the reason why we are all gathered here. And Balakrishnan Iyer is an avid Tamil scholar. And according to me, he's a personification of Sangha Surangam. You keep digging into him, Tamil and Tamil knowledge, and absolutely the most unique information will just flow out of him. So he's a personification of Tamil Surangam. And he's currently the honorary chairman of the Indus Research Center and the director of International Institute of Tamil Studies. And this he has taken up with passion after his retirement as an Indian administrative civil servant officer. So thank you so much for all the good work you do towards the Tamil community.
Thanks a lot. So nice of you to make this statement. It makes me more. Yesterday also I happened to attend the Chennai International Book Fair, where we had an opportunity to meet Mr. G. N. Devi with all his work. So when he mentioned about the JOC also, I felt really good because that a person like me who doesn't have any special qualification more than BA Tamil and MA Tamil, having all through worked in outside Tamil Nadu for 40 years. But then it makes me feel that we have spent our time worth. But let me tell you, I have nothing to, it's like already we have spoken in the form of question. But then I consider the JOC study circle a few years back when started in US, and then 2 rounds of discussion, particularly once involving the younger generation, the children coming and talking about it, various topics. I remember that sometime in Bhubaneswar airport, sometime in Vizag airport, sometime in Bombay airport, I used to own even once one from London also. So it was a phenomenal effort taken by US based Tamils, but it's a carried this particular book's message. But I am a great admirer of Professor Raj Mutharasan. When during my US visit, when I visited his place and met him, I still use this opportunity only to tell that a person like me, a career civil servant, and never had a lighter assignment, always having a stressful assignments like handling cyclone, handling flood, handling elections, that's a kind of job, and finance, budget management. With this only, I was trying to stitch my own experience. In fact, I've been talking about India as a rainforest, not a melting pot, not a salad bowl. Basically, all these findings are again and again trying to tell that plural foundations. We have been talking about the plurality in India is not, it's at a roots level. It's not a viludu, it's a where. It's a plural foundation. So that's what we have been emphasizing. And verupunarvillada porupunarvu, something like where you don't have a spread hatred. You talk about the inclusive language. The tribals of India and the tribals of Orissa taught me that. I used to wonder in late 80s when I learned something from the tribals. Even my own officers, they used to talk to them in a lighter vein, Adivasi. These people don't know much. That's a kind of little elite articulation among the officers. But then the tribal, the native intelligence, I used to wonder how much they had it. So that only made me to feel more humble and more rooted. And then it's basically my learning from the tribals of India. Their language, I used to learn Koya language. So now what people, the science, modern science talks, DNA studies talks, linguistics says, like people like Raj Mutharasan. I have a feeling that when people like him, because like Tony Joseph and all of them our friends, so they devoted their time and made a kind of narrative. But then bringing this multi-layered, transdisciplinary, putting it together, then I have a feeling Raj Mutharasan sir again and again telling he owes this to the society, and he must do that. And so I take this opportunity to make this request, which I have been often making to him over telephone. Recently, he addressed our recently he addressed our உலகத்தமிழ் ஆராய்ச்சி நிறுவனத்துடைய அறிஞர் அவையத்தில் we asked him to requested him to speak. He made a wonderful speech. We are looking forward for that day. Thank you very much, Subhadra, Bama, and all this. It's been the day is made. It's been a great. Thanks a lot.
Thank you, sir. Thank you so much. I call upon Sridevi for the vote of thanks.
Vanakkam. On behalf of the Journey of Civilization Study Circle, we sincerely thank Professor Raj Mutharasan Ayya for his clear and insightful presentation, and our own Balakrishnan Ayya for his thoughtful, engaging remarks. And we thank Subhadra for hosting and coordinating the program, and Arasaya for giving a warm support, and Mani Ayya for moderating the Q&A session. And we always, I especially thank Ms. Bama for her dedicated efforts in flyer preparation and speaker coordination, and for she does all the participation moderation through WhatsApp, and for the technical support and making the recordings and materials available. And even before Rajaya's presentation, she made it possible that she listed out all the YouTube videos from his previous talks, which I made it personally, and I went through all the videos to understand this deep subject, because it's really a different subject altogether. And finally, we thank all our Zoom and YouTube participants, both live and the future viewers, for your engagement and support, and waiting for so much hours listening to this wonderful session. Thank you all.
Thank you.
Thank you.
பாமாக்கு ஒரு ஓ போடலாமா? பாமாக்கு ஒரு ஓ போடலாம். பாமாக்கு ஒரு ஓ போடலாமா?
நீங்க முத்தரசன் ஐயாக்கும் பாலாயாக்கும் தான் போடணும். பண்ணதெல்லாம் வந்து கிளரிக்கல் ஜாப். அவங்க தான் இன்ஃபர்மேஷன்.
Okay, great, Bama. Thanks a lot.
Thank you, Bama. Thank you, Bama.
Thanks, Ma. Thank you.
Thank you, everyone. Thank you.
Yes.
Bye.
Okay.